one day. Why does it matter how much you slept? It does. It matters so much. So much, in fact, that there is a cycle behind this huge phenomena. A cycle so etched into our very existence that it's sometimes also called a biological clock. Circadian rhythms, CR. What is it exactly? Circadian rhythms are a rhythm in our body that play a fundamental role in the progression of cardiovascular events. Almost all cardiovascular diseases have a certain circadian misalignment, usually characterized by a change in metabolites. We will further explain this concept later, but we just have to understand reactions and their correlation with circadian rhythms for now. Still, all that is needed to be understood is the notion that your underlying reactions and factors influence the consequences. To best understand these consequences, we have to understand that in bioinformatics settings, it is not important just to ask the why, but the how, and later on, the when. But where do these biological clocks come from? A master clock located in your brain coordinates all the biological clocks in a living thing, keeping the clocks in sync. Think of these master clocks like a battery in a regular bedroom clock. These clocks consist of neurons, specified neurons, SCNs, that help regulate these bottom two levels, cellular clocks, which are found in organs and cells, and metabolites. Cellular clocks can be looked at as the hour hand in a clock. It regulates the minute hand, but it is regulated by the store manager, aka the SCNs, or the battery, whatever analogy you'd like to use. And the metabolites can be seen as the minute hand. It is regulated by this, but in the end, however many metabolites you have, for instance, let's say 60, that contributes to this, and all of these levels impact each other. Essentially, this is what the hierarchy would look like. The master clock, SCNs, comprise of 20,000 nerve cells that form a structure called the suprachiasmatic nucleus, or the brief word SCNs. The SCNs is a part of the brain called the hypothalamus, so it gets influences from the environment. They're just a bunch of specialized neurons. They're store managers. So where do metabolites plus circadian rhythms interact? Cardiovascular disease is highly complex. The process of establishing these highly complex thoughts involves metabolites primarily, an immediate or end product of the metabolism. To recap, master clock neurons primarily coordinate all of this, and they create these rhythms vital to an organism's physiology and behavior. The SCNs target and integrate light and feeding signals to entertain behavioral rhythms and regulate the cell's clocks in tissues and muscles. They're managers of cells, and they help form and regulate these cells and tissues. In this way, metabolic disease, like those found and caused through malnutrition, are always associated with misaligned circadian rhythms over time, resulting in many pathophysiological diseases like stroke, cancer, obesity, and type 2 diabetes, as well as hypertension. Hypertension, what is it? First of all, it's one of the most common cardiovascular diseases, and it is now characterized as an increase in high blood pressure, which is created by blocked arteries, which then make pressure on the arteries and have difficulty regulating blood. And you know that we need blood to survive. This is a crucial idea. SHRs, the rat model that I explained earlier, have been used to model hypertension and have been used to study, but the biomarkers found in these metabolomics studies were found at one-time intervals, meaning they were only looked at once. And that doesn't work because it ignores the notion that metabolites change over the day and your body changes. So many things happen throughout the day, as you would know. So if we were only to look at it once, we would be ignoring 23 hours worth of awesome data. So what now? The studies on biomarking, one of the most common cardiovascular diseases, are inefficient. So how do we look at them? The question is, we focus on the when. My proposal is not to treat the symptoms, but to diagnose hypertension by using circadian rhythms. If we study the effects on suprachiasmatic nuclei, SCNs, we can fully monitor the progress of circadian rhythms and uncover metabolic pathways, which control and regulate these cells that can contribute to diagnosing hypertension. So, I would like to find CR association through sample normalization, analyzing these samples, but also include LCMS, an analysis tool of chemicals, and separating them into liquid and solid. So you can analyze both and it's basically like mass spectrometry, which is looking at solids, but you're separating the liquids and the solids. I researched a study through which untargeted metabolomics technology was used to dynamically monitor changes in rats, not associated with hypertension as much. But in this study, they didn't include LCMS, and my goal is to include this here. It was completed to identify biomarkers in circadian misalignments. So afterwards, these concentrations were identified and normalized, but no LCMS. I would like to eliminate metabotypes as they are confusing and can't be understood in hypersensitive rats right now due to their still baby-like notion in the field. They're still pretty new. I would like to keep the SRR and WKI differentiation because it shows a differentiation between the two rats and shows changes and differences, which is key in metabolomics and bioinformatics studies. And I would like to add LCMS analysis to better sample and normalize the analyses that we get. So, what have I been doing? I stimulated an acetonitrile pathway in pathways because acetonitrile is one of the main chemicals used to separate the liquids and the solids in metabolomic samples, and as well as I found an ideal circadian rhythm intervention time. As you can see here, every 16-minute interval has 2 seconds to it. So therefore, 16 plus 16 is 32, and we can, from this graph, understand that every 32 seconds it would be an ideal CR intervention time in the case and the data that I put into here. So there's context gaps and questions, and what do we need to look at? Owing to metabolite movement throughout the day, clinical sampling of these chemicals sometimes can be inaccurate due to the variety of factors that poke, pull, and change these chemicals in your body's chemistry. The process requires SCNs, the master clocks. The reason? To verify clinical samples. When it comes to context gaps, it doesn't come to the why only, but the why, how, when, and more.